Hello, my name is Joris and I'm a program manager at StarFish Medical. And as a program manager, I'm often involved in development of new diagnostics. The world of diagnostics has changed dramatically over the past 5 years, especially because of the COVID-19 pandemic. We have seen literally thousands of products hit the market, and many of these products are designed to either detect antigens or through molecular testing to quantify how much COVID-19 virus is present. Because of all these products, what was novel 5 years ago is not novel anymore. So how does this new landscape actually impact developers of diagnostic devices? Well, actually that depends entirely on the core technology that's used. Let me give you two examples. Example number one, somebody wants to develop a lateral flow assay or a molecular test. These days, people would leverage a ready-made, pre-existent platform to onboard their core technology. So they are much faster in developing that first working prototype and testing for analytical performance and basically hitting their first critical milestones and pleasing their investor. So in the second example, a diagnostic developer would like to develop an electrochemical biosensor. Electrochemical biosensors are used to more accurately and more precisely quantify a particular target molecule. This is where lateral flow assays are, for example, not very capable. But there's also not a lot of pre-existing electrochemical biosensors out there. So where a developer would have to start is by pouring money into developing from scratch a whole platform and a device, first prototype, to do the first analytical performance testing and really proving out that technical feasibility. This means that investors would have to wait longer to see that technical feasibility as a first milestone, and where a developer would have more trouble getting to that milestone and getting a second round of funding. So how can we make the development of new diagnostic technologies more efficient? I think that we can learn something for what's been done in molecular testing and lateral flow assays. We should use a similar approach for other types of assays, where we use a modular system that allows a variety of different assay technologies to be onboarded. What I mean with this is that we get an assay that can be very quickly ported onto a consumable, and we can start testing. And we're testing for analytical performance. This is like, is the assay sensitive enough? Can users use this assay easily enough? And do we get other analytical performance factors that we're very worried about? This is really the first key technical milestone in the development process, and this is how we can do that much faster by not having to reinvent the wheel or developing something entirely from scratch. So what I mean by a modular system is that we've designed a cartridge that has all the fluidic paths and fluid control on it. In this fluidic control, we can also introduce a sample, we can introduce reagents, we can introduce lyophilized reagents. We have a particular window where we can look at cells or any biologics flowing past, and we can measure electric currents from the system. This allows us great flexibility in onboarding a variety of different assays. Please check out the blogs that we have online, or maybe drop me a line.